Hey everyone, welcome back to another video by Camroom 5. Today's video is also about the biology ATP O levels, where we are concerned about designing an investigation. So we're concerned about designing an investigation. So before you proceed about designing an experiment, you need to understand what's the objective. The objective is always to prove something. It is always to prove the importance of something or the effect of something, whether the effect is positive correlation or negative correlation. So to prove the effect or the importance of something. Every time you design an experiment, there are some steps that you need to follow, and I am going to make it very simple for you guys. For example, in step number 1, you mention what kind of specimen you are taking and how many of those. So you talk about how many experimental setups are taken. It means how many beakers, how many test tubes, how many plants, how many fruits, how many seeds. So you talk about the number of apparatus you're using, how many specimen you're using, if it's a plant, if it's an animal study, if it's a fruit study, what are you using, how many test tubes you're taking. You talk about that in the beginning. In step number 2, you talk about what apparatus are most important for your study. So what apparatus are needed. And you also hint about why they are needed. It is also good to mention why are those needed in the first place. Like for example, if you talk about a gas syringe, mention that I will be measuring some gas volume over here. If you talk about some test tubes, you do mention that you will be using it to collect your chemical specimen. In step number 3, which is very important step, you talk about how your experimental setups which you are taking are different. So talk about the different conditions created in each experimental setup. In each experimental setup. For example, let me give you an example. You can say that I have provided everything like water, soil, nutrients, fertilizer in test tube A. So test tube A has water, soil, seed, it has availability to heat or temperature, and it has availability to light also. But in test tube B, let's suppose you mention everything is the same but it's in dark. So here you are creating a clear difference, a clear contrast. Why are experiment A and B different? If you are taking another example, let's take another example. If you are saying that, for example, I want to study the effect of temperature, for example, to study the effect of temperature, let's suppose, on activity of amylase, You say that I have got A, B, C, D, E test tubes. You mentioned that everything has the same amount of water, same enzyme, same substrate, everything. But please mention that A will be at 10 degree, B will be at 20, C is at 30, D is at 40, and E is at 50 degree. You need to talk about how your various experimental setups are different from each other in step number 3. In step number 4, when you have talked about the difference, you need to mention what will you observe. Are you going to observe a color change based on what you're doing? Are you going to observe a plant growth based on what your experiment was? Are you going to observe some precipitates? Or let's suppose, for example, are you going to observe something else, some kind of movement? What are you going to see? Why are you waiting around the experimental setup? So you talk about that. In step number 5, which is the final step, you talk about how will you record your observations. Are you going to make a graph? If a graph, then talk about the axis also. What are you measuring on the graph? Are you going to make a histogram? Are you going to make a table? If a table, what will be the headings? That will make your experiment immensely structured, and that will help you gain good marks for such kind of questions. Let's do some past paper questions. Design an experiment you could carry out in a laboratory to investigate the effect of pH on the time taken for milk to coagulate using formase. Now the question is very clear and it's 4 marks. Effect of pH, number 1. Time to coagulate milk and using formase. So you have to talk about that effect of pH should be shown somewhere in the question. Let me show, yeah, like this. And time should also be mentioned in your question. You can start by mentioning that I'm going to take multiple test tubes because test tubes can collect milk. So you can say, take 5 test tubes to collect milk. When you say you are collecting milk, also give a hint about use a pipette to accurately measure the volume of milk. To accurately measure volume of milk. Now you have your milk. In each experimental setup, how are these test tubes different? So please mention that you can add multiple different pH values. So you can set these test tubes at varying pHs. You can give example like at pH is equals to 2, 5, 7, 9 and 12. This gives a good distribution from acidic to alkaline pHs. Then talk about how will you do it? By adding acid or alkali, and you can also use the word buffer because buffer is a chemical that maintains pH, so you can also talk about buffers. Then what are you going to observe? You haven't added the formase. So you can also talk about the idea that the same amount of formase should be there. So talk about add same volume of formase in each test tube. In each test tube, maintain same temperature. Maintain same temperature and allow the reaction. Now what did you have to measure? Remember you were looking at the time taken for the milk to coagulate. So talk about measuring the time taken for coagulation using stopwatch. So measure time taken for coagulation using stopwatch. You have to talk about the apparatus in each step if you can do that. How will you record your data? So you can say make a table with time values, and you can also talk about repeat the experiment and take means. Taking a mean or taking an average increases reliability, and this will give you a very solid 4 marks. Let me give you another example. Let's say another kind of question. This is also effect of pH. Increasing temperature, yes, this one is really good. So we can say design an experiment based on the method already described on page 5. You don't have to look at page 5, trust me. To investigate the effect of increasing temperature on the activity of catalase. Catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. And when oxygen is released, you can see some kind of fourth or some kind of foam coming. So the experiment in the question was actually that when you do the experiment, you could see some kind of foam coming on the surface. And the foam is because oxygen was released. So you had some foam, and that was page 5. Now, how many experimental setups will you take? So talk about that. So you can say take 5 test tubes. Label A to E. Now, what are you adding and how are you adding that? So you can talk about the fact that we will be adding hydrogen peroxide in them. So you can say add hydrogen peroxide. When you say hydrogen peroxide, aqueous, mention same volume using pipette or measuring cylinder. Talk about the apparatus. Your question is concerned about temperature, right? It's not concerned about the volume of hydrogen peroxide, so same should be there. You can also mention same volume of catalase solution be added. So you're also adding catalase over here. Then mention that concentration of both should be same, because concentration are also very vital here. So concentration should be same in each test tube. How are they different? Because step 3 is about how are they different. So set each setup on different temperature using thermostatically controlled water bath, using thermostatically controlled water bath. Now, when you have talked about it, do mention what kind of temperature range are you talking about. Make it more concrete, like 20 degree, 40 degree, 60 degree, 80 degree, and 100 degree. Then say, what are you measuring? So talk about see the foam develop after same time, because time should also be same. Then talk about measure the foam and record. Measure the foam volume and record on table. This will also give you like complete solid 6 marks. I want you guys look at some more past paper questions, because I have many and I have planned on doing another video on it. Stay tuned, guys. Thanks.